We're going to cover some more ground here in the immune story, and this concept that I'm going to be talking about now, it is, it looks scary, but it's not, but it's somewhat challenging to explain what's going on there. So we're going to be talking about the proteins of major histocompatibility complex. Okay. So essentially, those are the special proteins expressed on the surface of the cells, which role is to identify a cell as self or non-self. Is that clear? So let's say, let's focus on Mhc Class 1 first. Okay, so here's the Mhc Class 1. These proteins are expressed on all nucleated cells, from hepatocytes to B cells to neurons. You with me so far? So what they do, your cells synthesize proteins. Do you agree with me? They produce proteins all the time. Does that make sense? The fragments of those proteins that are normally produced in your cell, they are exposed on the surface here. This is called antigen presentation. So basically, Mhc Class 1 constantly, these molecules constantly wave the flag saying, I'm fine. Do you understand what I'm trying to say? So say your liver cells waving the flag saying, hey, we're not infected, we're not malignant, and we are self. So when Mhc Class 1 presents self-antigen, your immune system passes by, doesn't respond to it. Is that clear? Now when, say, cell is infected by intracellular pathogen, say a virus, it starts to wave a different kind of flag. Instead of self, it starts to wave something non-self. Does that make sense to you? So that means, hey, I'm infected, I'm not myself anymore, kill me. Clear? Now the unique cells, called antigen presenting cells, or dendritic cells here, they can actually present, dendritic cells are basically phagocytes, okay? They're like very specialized macrophages, and their function is to engulf a pathogen, break it down, grab a fragment, the antigen, and wave it outside. But in this case, dendritic cell will be waving. They don't show Mhc Class 2, but say that's Mhc Class 1. It will be waving the antigen to the rest of your immune system, saying, hey, this is what you got to look for. When you go out there and you are in the circulation and you float around, this is the antigen that you're looking for in other body cells, like in the hepatocyte. We'll get back to this idea of antigen presentation a bit later. Now, Class 2 is the type of major molecules of major histocompatibility complex that are expressed on macrophages, dendritic cells, and B cells, so-called antigen presenting cells. And we will talk about the different roles between Mhc Class 1 and Mhc Class 2 a bit later. Now, on the very basic and conceptual level, I want to show you what dendritic cell does in relation to other immune cells. Let's say Lauren here, she is an immune cell, a T cell, okay? We clear? She's a T cell of any sort. If she sees that antigen individually, she doesn't respond to it. She needs to be presented with the antigen. So some other cell, in this case me, dendritic cell, will have to show the antigen to T cell saying, look, this is something bad. So you have to mount a response against this. We're clear. The mechanisms through which MHC type 1 and MHC type 2 work is slightly different, and we'll address this. But so far, does that make sense to you? That this is the antigen presentation. That's really important. T cells have to go through antigen presentation in order to start working. We're clear? Good. So let's talk about what happens during the T cell maturation. T cells originate in the bone marrow, and then they migrate into thymus. In the thymus, they mature, forming two main populations of T cells: CD4 T cells and CD8 T cells. The fundamental difference between these two populations is that they produce different functional types of effector cells. CD4 cells have class 2 MHC proteins on the surface, and therefore can recognize only class 2 MHC on the dendritic cells. Am I clear? So how you remember, I have a mnemonic rule. It's always 8. So CD4 recognize class 2. 4 by 2 equals 8. CD8 recognize class 1. 8 by 1 again equals 8. So far, is that clear? Now, this is something really unique and kind of at the level of a law. CD4 cells always recognize class 2, while CD8 cells always recognize MHC class 1. This is referred to sometimes as Mhc restriction. No exceptions to the rule. If it's CD4, it's going to recognize MHC class 2. If it's CD8, MHC class 1. Does that make sense? So once these cells, CD4 and CD8, mature, they enter the circulation. They enter the lymphatic system. So here at this stage, cells are in the lymphoid tissue, such as the lymph node or spleen. And in the lymphoid tissue, T cells are presented with the antigen. Now I want to remind you that MHC class 1 is recognized exclusively by CD8. Can you see that? Can you see that recognition? All right, do you see that? So CD8 cell learns about the antigen that it has to respond to via presentation by MHC class 1. Now these proteins, MHC class 1, where are they expressed except the dendritic cell? Where else? MHC class 1, where else they are expressed? It's right there. Class 1 MHC. Where are they expressed? All nucleated body cells. What does that mean in terms of the immune response? It means that CD8 cell right here, when it gets activated, it enters the circulation and it can recognize that same antigen presented by Mhc class 1 on any body cell. Let's say the dendritic cell presented the hepatitis C antigen. So CD8 cell will enter the circulation, travel to the liver, see the same antigen bound to the same Mhc class 1 molecule and say, aha, I gotta kill it. We'll kill it. Does that make sense? Imagine that we have a different situation. We have a person with systemic tuberculosis. So this person's CD8 cells, when they recognize the antigen, they proliferate it, differentiate it, they enter the circulation, they go to the bones, see osteocytes infected, or osteoclasts infected with mycobacteria, say, aha, I'm going to kill it. Go to the brain, see microglial cells infected with mycobacteria, say, aha, and they say that aha in every part of the body where they see the antigen presented by Mhc class 1. Does that make sense? So, and I used several times the word they kill it. They kill it. It turns out that CD8 cells, when they differentiate, they become cytotoxic T lymphocytes or CTL or killer cells. Sometimes they simply called T killers. Now what about CD4? Well, CD4 cells can only recognize the antigen presented by Mhc class 2, which is found on a very limited number of cells. Macrophages, dendritic cells, and B cells. So what do these cells do when they are presented with an antigen? Well, it turns out that CD4 cells, after the activation, differentiate into helper or regulatory T cells. So they don't kill. They help other cells. Does that make sense? Now, before we move on to how CD4 cells actually do that, I want to mention a few words, a few things that happen to basically all T cells in the thymus. So think about this. Remember we talked about B cells, right? B cells, all they have to do is not to recognize our own antigens. Is that clear? So they go through negative selection. Bone marrow makes sure that we have no self-reactive B cells. In case of T cells, we have one more condition. T cell, on the right, must be able to recognize MHC. Must be able to get presented. So basically, what I'm trying to say, if Oliver is T cell, if he does understand that I'm showing him an antigen, he's good. If he has no clue what I'm doing, it's not a functional T cell. Does that make sense? Now, that ability to recognize MHC, you sometimes call it being fit for the job. Okay? So that ability is selected during the process of positive selection. All cells that can't, like this cell here, okay, this cell cannot recognize MHC, that leads to apoptosis. And then, during negative selection, T cells that recognize self-antigens, they go in apoptosis. Does that make sense? So that, call it, don't go berserk. If basically negative, so here's what happens. If positive selection fails, you have a bunch of T cells that are totally dysfunctional. Makes sense, right? They just can't be presented with antigens. They're useless. If negative selection fails, you have a bunch of T cells that will recognize their own tissues and will attack them. Got it? So basically, in the first case, you have an immunodeficiency. In the second case, you have autoimmune disease. Now, what T cells can do? So what happens during activation? So that's your helper T cell, right? And you see that helper T cell recognizes the antigen, that tiny little red dot, presented by class 2 Mhc. And it differentiates into helper cells and the memory cell. Okay, we're good? Now, what are the function of this? So I'm going to overview the function of CD4 cells, and we're going to wrap it up. So, first, memory cells. Understood the function? So, memory T helper cell remembers the antigen like stored for the future use. Okay, cross it over our list. Next, remind me please, which Mhc class does T helper CD4 cell recognizes? Which class? 2. And Mhc class 2 are expressed on dendritic cells. They presented the antigen. And also on macrophages and B cells. Does that make sense? So, turns out, when T helper cell here recognizes the antigen presented by B cell, it activates B cell, leading to differentiation to plasma and production of antibody response. Remember, we talked about B cell activation, and I told you that it can be T cell independent and dependent. This one is T cell dependent activation. Hold on, I'm going to give you an analogy that I really like. Another type of helper cells will recognize the antigen presented on class 2 Mhc on macrophages, and it will activate macrophages and direct them against that particular antigen. On top of that, some of those helper cells will participate in the activation of CD8 cells, of cytotoxic lymphocytes. Does that make sense? And will help to activate them so they can kill other cells. Now, the special population of T helper cells that I want to mention, regulatory, Tregs. Instead of stimulating the defense response, they actually dampen it. Why? Why do we need to dampen? Why do we need to lower the immune response? Should we just let it go forever? It's probably a lousy idea, right? Because immune response generally is damaging tissues. Okay? So T regulatory cells, they actually decrease the immune response and kind of control it. And it was shown that T regulatory function of these cells is reduced in autoimmune diseases. For instance, T regulatory cells are functionally defective in IBD, inflammatory bowel disease. Now, the analogy that I use for explaining what CD4 T helper cell does, it's the football coach. You know, if you take a regular football coach, whether it's a high school coach or professional NFL coach, if you will try to put them on the field, that's going to be laughable. Right? They cannot run, they probably can't throw very well, but they work from the sidelines. They tell quarterback, say CD8 cell, what to do. They tell wide receivers, the B cells, what to do. They tell linebackers, the macrophages, what to do. And if you remove the coach, the whole team falls apart. And there's a disease in which you remove the coach. It's called HIV. In HIV, the only cell that is gone, the only cell, well, type 1, HIV type 1, let's put it this way. If CD4 cell is gone, right, in HIV, then there is no T cell dependent activation of B cells. There is no macrophage activation. And cytotoxic lymphocytes are suffering. So canceling out just one cell type basically cancels out the entire adaptive immune system, or 90% of it. Makes sense, right? Just one cell type, and the people become terribly immunodeficient. Does that make sense functionally? What helper cells do? So, like, arguably, they're probably most important cells of the adaptive immune system, although they don't kill anything, they just trade. Okay? They just help other cells. Got it? Okay. So, we're going to wrap it up. I'm going to meet you on Wednesday.